Hello guys, welcome to my channel. So as you know, we are studying about the cell cycle control system. If you saw the part 1 and part 2, we did the introduction, we did the G1 phase, that how the cell cycle control system controls the cell cycle if DNA is damaged in using different mechanism. So now moving to the S phase. So what happens in the S phase? Before cell divides, it must replicate its DNA, right? So this replication must occur with extreme accuracy to minimize the risk of mutation in the next cell generation. So, and of equal importance, every nucleotide in the genome must be copied once. It should be copied only once in the genome, not twice. So only once, to prevent the damaging effects of gene amplification, right? So how this cell cycle control system initiate DNA replication and at the same time prevents the replication from happening more than once per cycle. So as we know here, for the movement from G1 to S phase, there is SCDK, that is our cycle-independent kinases, right? So what happens in this stage? So like any monumental task, so the configuration chromosome for application requires a certain amount of preparation. Like, so as we know, there are two chromosomes, right? Like this chromosomes are involved for when the DNA. So we'll study the DNA replication in after we finish this cell cycle control system, but for now, right? So just remember, the chromosome for replication requires a certain amount of preparation. So for eukaryotic cells, this preparation begins early in G1. So like before the DNA replications begins in S phase, so the preparation is already started in the G1 phase. So when the DNA is made replication ready, by the so what happens in the end of the G1 phase? So first, all right, let me remove something. Remove this. Maybe this much will be enough. So I'll write down here as phase. All right, so it will start, the DNA replication preparation will start at the G1 phase, and what happens in the G1 phase is the at the end of the G1 phase there is a recruitment of protein. So okay, let me write it. End of G1 phase. So what will happen at the end of the G1 phase? So recruitment of proteins to the site along each chromosome, right? There are two chromosomes, so there will be a recruitment of protein along each site of the chromosome where the replication will begin. So for example, like this is chromosome, like this is a site. I will write down site of replication. So this will be the preparation, like there will be a recruitment of protein for each of this chromosome, right, where the replication will begin. And this nucleotide sequence, so it will be a nucleotide sequence, of course, right, for the DNA, as we studied in the introduction of DNA. So what it's called? So the recruitment of protein, and that protein is a nucleotide sequence, and this nucleotide sequence is called origin of replication. Is called, okay, let me know. Nucleotide sequence is called origin of replication, right? Serves, and this origin of replication of this protein serves as a landing pad for the proteins and the protein complexes that control and carry out DNA replication. So this will be serve as a landing pad. All right, okay. And one of these protein complexes is called as the origin recognition complex, right? So one of the protein complexes, right? So these complexes that are being used from them, one of them is called an origin recognition complex, right? So ORC complex, and in short it's known as ORC. And this ORC remains perched at the top the replication origins throughout the cell cycle, and this will remain atop, right? So what happened in the first step, the replication initiation, the ORC results a protein called CDC. So let me draw something maybe. All right, so we'll draw here. For example, what we'll do, so like, and no, I'll just draw. So for example, this is our site, right? And here protein, we'll use this. It's also not that dark, but so this, the round circles are the proteins that are, so once at the end of this side there will be recruitment of protein right at this side, and this is the origin of our replication protein, the nucleotide sequence. And in one of this protein complexes, this one for example is OR, say we'll make a star here for the ORC, that is our origin recognition complex, right? So in the first step what will happen, the ORC recruits a protein that is called CDC. So what will happen, this ORC, like, so this will be for example our first step, and this is our ORC. So this will recruit a protein. I will write down here. Oops. Recruit protein, and that is called CDC6. CDC6, right? This CDC6, what will happen? This CDC6 concentration rises early in G1 phase. So this CDC6 concentration will start rising early in the G1 phase. So I will write down here in the bracket, concentration rises in early G1. Concentration rises in G1. So what will happen? So there will be a recruitment of protein, right, at the site of the replication, and the nucleotide sequence and the protein complexes are known as the origin of replication, and one of the protein complex is known as a origin recognition complex. So this ORC start recruiting a protein that is a CDC6, and the concentration will rise early in the G1 phase of CDC6, and together this CDC6 and the ORC, so like I will just draw a simple, like here for example this is our ORC, and this recruits CDC6 maybe like this shape. I will put CDC6 here. So this will join ORC. So this together protein CDC6 and ORC, what it will do, it will start opening the DNA helix together, right? And it will become ready for the origin of replication. So this will act on, this both will act on DNA helicases, and it will open up the DNA helicases, right? And it will be ready for the origin of replication, and it's known as the pre-replicative complex, and this will be in the place and replication origin is loaded. So this together act on DNA replication. So for example once they opened up the DNA, all right, let's put it here. This is our ORC, and for example, this is our DNA pre-replicative complex. I will write it down. Pre-replicative complex, right? So this, the DNA, both they separated both DNA helices, and in perfect sequence, this is ready as a pre-replicative complex. And what will happen? So after doing this, the CDC6 will be separated from the ORC. All right, and this will be ready for our replication origin, right? And it's ready to fire. So the signal to commence the replication comes from SCDK. So after this, so this process is done here at the end of the G1 phase, and when it enters into the S phase, so this is stimulated by SCDK, and once it comes here, the signal to commence the replication comes from SCDK, the cyclin dependent kinases complex that triggers the S phase, right? So SCDK is assembled and activated at the end of this G1, and during the S phase, it activates the DNA helicases. It will activate this DNA helicases that is in a pre-replicative complex and promotes the assembly of the rest of the proteins that form the replication fork. We'll study DNA replication, but it promotes a replication fork, and in doing so, the SCDK essentially pulls the trigger that initiates DNA replication, and then how the DNA is replicated and it moves further in the phase. Alright, so this is the how SCDK helps in DNA replication. But SCDK not only triggers the initiation of DNA synthesis at a replication origin, it's not only the function that is just trigger this, but it also helps preventing re-replication. Like it's not in this cycle, like again the DNA is replicated and like there are like two DNA replication now. What it does so helping by phosphorylating this. So this SCDK will trigger this, and SCDK also phosphorylate CDC6 protein. Once the CDC6 protein is there, its function is done, right? So it will be separated and it will be phosphorylated by the CDC6, which marks that protein for degradation, and eliminating the CDC6 helps ensure that DNA replication cannot be reinitiated later in the same cycle, right? So this is how SCDK does. DNA replication initiates and stops being re-replicating twice in the same cycle, right? Right, so this is how DNA is replicated, and earlier we see it when the DNA is damaged, right? In how the cell cycle control system uses its mechanism to delay the progress through G1 to S transition, preventing the cell from replicating damaged DNA, right? But what if the errors occur in the DNA replication, right? What happens like if the DNA is replicating in the S phase and like there is some error? What happened? How it is stopped from going further to G2 or M phase? So if you remember when we discussed the introduction, I guess, in that we discussed the activity of MCDK, right? How MCDK is activated. So MCDK is inhibited first by phosphorylation at particular site. For example, like here I draw the picture that I removed it, but okay, let me see. So for example, this is our CDK, and right, we like draw it like this, and this both together is MCDK, right? But this is still not active, it is still inactive, and this is actually phosphorylated together, right? This both are phosphorylated together, so it remains in inactive state, right? For cell to progress in mitosis, this inhibitory phosphate must be removed. So this, okay, so I will show you. So, right, so we'll discuss like this, right? CDK and our kinases. This is our kinase, protein kinase. Oops, I don't have space, but for example, kinase and CDK, and together it becomes MCDK. As it is phosphorylated, this MCDK is phosphorylated with, for example, it's actually known as V1 protein kinase. It is phosphorylated by WEE1. This is a phosphate kinase that is known, and this is but still inactive after phosphate. So to become active, it should be actually phosphorylated, means dephosphorylated. So right, so in order to become active, so they have to remove this, we have to remove this inhibitory phosphatase, inhibitory phosphatase. So how it removes? It removed by a phosphatase known as CDC25. CDC25. So this will act on this MCDK, it will remove the inhibitory phosphate, and then the MCDK will become active. So that will progress through the cycle. But if the DNA, if the replication has any error, the DNA replication has any error, what will the cell cycle control system do? It will inhibit this CDC25, preventing the removal of the inhibitory phosphatase from the MCDK, and as a result MCDK remains inactive and the M phase is delayed until the DNA replication is complete and any DNA damage is repaired. Right? So this is how it stops. Right? So now we studied S phase, so now we'll study the M phase. So it's this whole process will S phase, then G2 phase, and then it will be M phase. So I will make some space, so we don't need this, so we'll just leave. Alright, so let me blast him as well. Alright, so M phase, the most important phase. Right, so although the M phase, so that includes mitosis as well as plus the cytokinases, occur over a relatively short amount of time, about 1 hour in a mammalian cell that divides once a day or even once a year. It is by far the most dramatic phase of the cell cycle. So during this brief period, the cell reorganizes virtually all of its components and distribute equally them into the two daughter cells. Right, so the earlier phases, the G1 S phase or the G2 phase of the cell cycle, in effect serve to set the stage for the drama of the M phase. The central problem of a cell in M phase is to accurately segregate this chromosome that were duplicated in the preceding S phase, right? So that each daughter cell will receive an identical copy of a genome, right? That is the most important thing of the cell cycle, right? So as we know, the MCDK drives entry into the M phase and the mitosis, right? So that is our in M phase, the MCDK, that is the driving point or the entry point into M phase. So one of the most remarkable features of this cell cycle control system is that a single protein complex MCDK brings about all the diverse and intricate rearrangement that occurs in the early stages of mitosis. Right, among its many duties, MCDK helps prepare the duplicated chromosome. This also helps prepare the duplicated chromosomes for segregation and includes the assembly of the mitotic spindle. What is mitotic spindle? The machinery that will pull the duplicated chromosomes apart, right? So how it does the MCDK complex, as we know, right? As we discussed here, it's accumulate throughout the G2 state. During the G2 state, this MCDK will start accumulating, but this stockpile is not activated until the end of the G2. And when activating, it is activated by this CDC25, right? Phosphatase removes the inhibitory phosphate holding this MCDK activity, right? So once it is removed, MCDK is active, but this activation is self-reinforcing. So once activated, each MCDK complex can indirectly activate additional MCDK complexes by phosphorylating and activating more CDC25. And this activated MCDK also shut down the inhibitory kinase, this V1, this we studied right now. So this is an inhibitory kinase, right, that attaches to MCDK and it becomes inactive. So it shut down this as well, right, further promoting the production of activated MCDK. And the overall consequence is that once MCDK's activation begins, it ignites an explosive increase in MCDK activity that drives the cell abruptly from G2 to M phase. So when the cell enters the M phase, the duplicated chromosomes condense become visible under the microscope as a thread-like structure. So the protein complexes condense in, that is responsible for condensation. Complexes called condensons. Right? This helps carry out this chromosome condensation, which reduce mitotic chromosomes to compact bodies that can be more easily segregated within the crowded confines of a dividing cell. So the assembly of condensin complexes onto the DNA is triggered by the phosphorylation of condensins by MCDK, right? So even before mitotic chromosomes become condensed, the replicated DNA, that the DNA is replicated, is handled in a way that allows a cell to keep track of the two copies. Immediately after a chromosome is duplicated during the S phase, what happens? The two copies remain tightly bound together. So once it's replicated, so there will be two copies. So like for example, so this is how it is actually. Two copies, right? These two copies remain. What will happen? The two copies are generated, and these two copies are remain tightly bound together. These identical copies, these are called sister chromatids, right? And each contain a single double-stranded molecule of DNA along its associated protein. And this both sister chromatids are held together by a protein complex called cohesin. So for example, this is held together. So this is our cohesin. All right, I'll write it down. Sister I held together by protein complex called coesins, right? So this is coesins, and this and the bulged part is actually condensins, right? So this coesins assemble along the length each chromatid as the DNA is replicated, and this is how it is held together. So this cohesion between the sister chromatid is crucial for proper chromosome segregation, and it is broken completely only in a late mitosis stage to allow the sister to be pulled apart by the mitotic spindle. And defects in any of this sister chromatic cohesion lead to major errors in the chromosome segregation. So in humans, such missegregation can lead to abnormal number of chromosomes. If this is not properly segregated, it will be missegregated and it will be resulting in abnormal number of chromosome as individual with Down syndrome. Individual with Down syndrome who have three copies of chromosome 21. Oops, sorry, 21. Right? So cohesin and condensins are structurally related and both are thought to form a ring structures around a chromosomal DNA, but whereas cohesins tie the two sisters, right, chromatids together, condenses assemble, so this ties together, but condenses actually assembles on each individual sister chromatid as a start of M phase and help each of these double helices to coil up in a more compact form. Right, together this protein helps configure replicated chromosomes for mitosis. Okay, so this is how our two sister chromatids are held together, and then it's in the M phase to pull apart and each will be given to the daughter cells. Right, so the next we are studying M phase. So then the M phase, the first one is the mitosis. That's again a whole entire cycle, right, for the separation of the two cells. Right, so I think we'll make one more part for this, a separate video for mitosis during the M phase. Alright guys, thank you guys for watching and don't forget to subscribe the channel. We'll see you in the last part of this video, that is mitosis.